Hi guys, today we're going to be talking about cystic fibrosis. So cystic fibrosis is an autosomal recessive genetic disease that is inherited. Cystic fibrosis specifically affects the CFTR gene, or the cystic fibrosis transmembrane regulatory gene. That gene is located on chromosome 7. It codes for the CFTR protein, which is primarily responsible for chloride passage from the inside the cell outside the cell. Now, CFTR is primarily found in epithelial cells, and its primary function is to help water down mucus secretions. Okay? Delta F508 mutation is the most common one, and that's why we're going to be talking about that one today. And when that mutation is in effect, it leads to thicker and more viscous mucus production from the epithelial cell. CFTR protein is made up of many different domains. Starting off with the amino terminus, we have the transmembrane domain 1 made up of alpha helices. Moving on from that, we have the nucleotide binding domain 1. And moving from there, we have the regulatory domain. Moving from there, we have another set of alpha helices making up transmembrane domain 2. Moving from that, we have another nucleotide binding domain called nucleotide binding domain 2. Moves on to the PDZ protein, which is attached to the cell's cytoskeleton. Moving on to that, we have our end terminus. All right, so the way that this is activated is it's going to be activated through phosphorylation by the protein kinases, two specifically, PKC and PKA. PKC must phosphorylate the regulatory domain first, open up the site that PKA is going to phosphorylate. Once that is phosphorylated, it can move on into attaching ATPs to its nucleotide binding domains. Once those are formed, then it is activated, allowing chloride to move through. So before we continue on, I'd like to first discuss how these two are activated. Okay? To go do that, we need to move to our 7-transmembrane or G protein coupled receptor pathway. Okay? This pathway works very similarly for both. It starts off with a ligand, which can be epinephrine or acetylcholine, just as examples. Those move into the receptor site, bind, which then goes over and causes the alpha S, which is attached to GDP, and also a beta and gamma subunit. It's inactive in this form when it's attached to GDP. Causes the GDP to be spliced off of the alpha S subunit, which then binds to GTP. Once this is bound to GTP, this whole subunit breaks off from these alpha helices and also breaks off from the beta and gamma subunit and is activated and moves over to adenylyl cyclase, which then takes ATP, forms it into cyclic AMP. Cyclic AMP then is used to bind to the regulatory domain PKA, which then allows it to be activated, move over into phosphorylation. Moving onto PKC's activation, we have a very similar thing where the ligand, like the ones we discussed earlier, come in and bind to the receptor site, thus cleaving off GDP, allowing GTP to be added on, which then removes it from the alpha helices and splits it off also from the beta and gamma subunits and moves over to PLC. PLC is a phospholipase C protein, which takes PIP2 inside the membrane and converts it into DAG, which is diglyceride, or IP3. Now these two split off and do separate things. IP3 moves over to the endoplasmic reticulum, and it moves over to an IP3 receptor, activates it, which allows calcium within the ER to be moved out into the intracellular environment. DAG then moves over and binds to PKC, allowing it to be activated and phosphorylate the regulatory domain on CFTR. Once those are phosphorylated, it's going to allow a single thing of ATP to bind to one of the active sites on NBD1. Once that is bound, NBD2 is going to come over and allow this active site to bind to that same ATP, forming a heterodimer. Once that heterodimer has been formed, a second thing of ATP is going to come in, this active site on NBD2, and bind. Once that's been bound, that's going to allow a conformational change in NBD1 to bind to that second thing of ATP, creating a full heterodimer bound to 2 ATP. Now this one right here is a fully activated CFTR protein. All right, so now let's talk about the ion movement in epithelial cells when CFTR is activated. First off, an activated CFTR will inhibit a channel, a sodium channel called ENaC, which is epithelial sodium channel, which prevents sodium from moving inside the cell from the extracellular space. ATP will also allosterically inhibit that as well. CFTR will then come over and activate this thing over here called ORCC, which is basically just another chloride channel, allowing more chloride to move from the intracellular space to the extracellular space. We now need to compensate for the fact that we're not getting as much sodium inside the cell as we were before. That leads to the activation of the sodium-potassium chloride channel, which allows 2 chloride ions in every 1 sodium and 1 potassium ion. Now, the sodium and the chloride are being compensated for by these other channels that are being activated. But potassium needs to be able to leave somewhere. And that's where these other particular activations come in play from these 7 transmembrane proteins. For PKA, PKA will activate a cyclic AMP-dependent potassium channel, allowing potassium out. IP3 will activate the ER to release calcium. And calcium will come over here and obviously activate this one here, and then move over here and activate this other potassium channel, which allows more potassium to. All of this here sums up to a overall negative charge forming on the apical side of the epithelial cell and a relatively positive charge on the basal side. This separate things in charge makes it so that the ENaC channel, which usually takes sodium extracellular to intracellular, will flip and move across the membrane into the extracellular space, making NaCl, or sodium chloride, over here. Now, this change in concentration draws water all the way over into the extracellular space via osmosis to offset that concentration gradient. This water moving over here essentially just becomes salty water, which in cystic fibrosis patients helps to thin out the mucin that gets produced in the mucosal cells. Okay, before we move on, I'd like to talk a little bit about the delta F508 mutation. Now, this mutation is a deletion mutation, which specifically removes 3 organic base pairs from the CFTR gene. These base pairs code for the phenylalanine amino acid on the 508th position of the CFTR protein. That's the F508, right? Phenylalanine over there. The deletion of phenylalanine affects the NBD1 structure. Interestingly enough, the deletion has only a minimal effect on this CFTR protein function. Why on earth is it an issue then? Well, the issue is because ER has regulatory mechanisms in place to stop mutated proteins from taking hold. Well, it sees that this is mutated, it marks it for destruction. As a result of this, in CF patients, only about 2% of the CFTR made make it to the cell membrane. This makes sense when you think about it in terms of the lungs, because you have a very small amount of CFTR, creating that ion concentration gradient to draw water over, meaning you're going to get a small amount of water compared to the large amount of mucus buildup being created by the mucous membranes.